In this section, we will talk about the Calvin cycle. And the main idea of the Calvin cycle is that the CO2 is reduced to produce sugar. And I'm going to, as usually, show you a video of your textbook that's displayed in a very pretty way what is the Calvin cycle and all the different reactions that are taking place.
The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.
Now I'm going to recap a few ideas that they can be in the test. First one, remember that the ATP and ADPH, these two molecules power the sugar synthesis in the Calvin cycle. So in this schematic, I show you that there is an input and output. So the input are like the ingredients of the Calvin cycle. The ingredients are CO2 that come from the atmosphere, the ATP and NADPH that come from the light reactions, and here they combine with the rubisco to produce the Calvin cycle, and as an outcome, as an output, you have this energy-rich molecule that is a 3-carbon sugar molecule called glyceraldehyde 3-phosphate. And in plants, this G3P is used to produce different organic molecules such as glucose and other ones. So let's recap. The second thing that I told you is that the Calvin cycle makes sugar within the chloroplast. The third thing that I told you is that in order to produce sugar, they have to have some ingredients, and these were word ingredients: CO2 that come from the atmosphere, and ATP and NADPH that are generated by the light reactions. And the fourth one is that the Calvin cycle uses these three ingredients to produce a 3-carbon sugar that is called glyceraldehyde 3-phosphate, or G3P. And finally, that a plant, they may use this G3P to make glucose and other organic molecules. And here there is a question that I'm sure that you can answer. So reactions that require CO2 take place in, so read it, A, B, C, D, E. You can stop the video for a second. Okay, I'm sure that you have the answer already. We just studied it. We have this schematic, and I show you that here were the ingredients of this process of the Calvin cycle. So here's the answer. The Calvin cycle alone will require to have CO2. The light reactions will require water and light, and the light will break the water into OH and hydrogen. So very easy. So it is B, and let's go to the next slide. In this slide, I want just to use again this schematic of your textbook. to talk about now the Calvin cycle and reaction, that they are also called dark or carbon reactions. And there are 5 things that I want that you remember. The first thing that I want that you remember that the Calvin cycle are light independent. Yeah, they can occur in light or dark. The second thing that I want that you remember is that they are produced in the stroma of the chloroplast. The third thing is that they use, as we said, this ATP and ADPH coming from the light reactions to turn the CO2 and reduce it into a carbohydrate. And finally, the fourth one from this slide is that the CO2 enter to the Calvin cycle to produce a sugar, as we said before in the previous slide. Finally, the last thing that I want that you remember, there is a very important enzyme here that have 5 carbon that is called rubisco and is in charge of catalyzing the first reactions of the Calvin cycle. So 5 things that I told you, please, very general, but they can help you for the test. Now we're going to talk about the different phases of the Calvin cycle, and in your textbook they focus on 3 phases. There are many more enzymes and metabolites, but for this class this is enough. In biochemistry you will see many more intermediate processes and many more enzymes. So as you watch in the video, there are 3 phases. In the first one there is a fixation of CO2, so the CO2 enter and mix with the rubisco. They interact together and you start with 1 carbon molecule, and as a result of this interaction between the rubisco and this CO2 molecule, you will have a 3 carbon molecule in the next stage, that is the reduction of the 3-PGA. Then later you will go and try to regenerate the rubisco from G3P and you come back to a 5 carbon molecule. So let's see this with a schematic. Here you have 1, 2, and here 3 phases. In the first phase, as we said, there will be CO2, then it will interact with the rubisco. This one has 5 carbons, this will have 1 carbon, and as a result of that you get this molecule that has 3 carbons. So you will get, for example, 2 molecules of 3 carbons, and then the next step is to get this glyceraldehyde 3-phosphate that we talked before, and as a result of that you can produce sugars. And the next step is to regenerate the rubisco enzyme. And so we have, in the beginning we have the fixation of CO2 until the formation of this 3-PGA. Then in the second one you are going to reduce the 3-PGA and produce glyceraldehyde phosphate, and as a result you produce sugar. And in the last step is the regeneration of the rubisco, and the process can start again. In this slide I want to summarize what happened with the energy molecules during the Calvin cycle, and you have 2 molecules that are important, that is ATP and NADPH, that come from the light reactions, and you will turn those molecules into ADP and NADP+. And from this one you will require 9, and here you will require 6, and 6 of them, and you will spend here 6 of ATP and 6 of NADPH, and here you will require 3 more of ATP. So let's summarize what I just told you. So you will require 9 molecules of ATP and 6 molecules of NADPH, and they will be spent in the beginning in the reduction of the 3-PGA molecule. So 6 ATPs will turn into ADP, and in a similar way the NADPH will turn into NADP plus, and later in the regeneration of the Rubisco you also spend 3 molecules of ATP. Okay, with this schematic I want that there is no need that you memorize this. No, there is no need, not in the test. Yeah, I'm not going to test and you need to remember all these different things, yeah, all these different steps. What I want to tell you is that this is what you're going to learn in biochemistry, that this process is much more complex that I'm showing you and that your textbook is showing you, but I want that you just focus on few things and you learn it well, and later if you get interested in this topic in biochemistry you will study it in much more detail. So I just told you here that there is CO2, that there is a Rubisco enzyme, and they interact and they produce these 3 phosphoglycerate, and they need to spend ATP and ADPH, and they have different products, and at the end you end up regenerating the Rubisco by spending more ATP. Okay, so again, this will be not in the test. This will not for testing. You don't have to memorize this. I just want to show you that this is more complicated that I'm explaining it right now. And again, go to your textbook. And in your textbook, this is what they show, a much more simplified version. Just to finish the class, in your textbook, they also show you very pretty schematics that they show you that the CO2 enter to the Calvin cycle, and it goes from 1 carbon through 3 carbons, and then it goes through 3 carbons, and then it goes back into 5 carbons. So go, and if you want, give a read to that. Also, this is a summary of the different phases according to your textbook, and again there are 3 phases: fixation, reduction, and regeneration. And here they show how much energy you are spending in each one of those. Okay, I don't want to be more repetitive, but if you have any question, just let me know and we can talk about that in class. Have a good day.